We're glad to be here. Glad Larry and Jake and his crew invited us. Short drive for me. I live in Carroll, Iowa. About an hour and a half. Known Larry probably, how long, Larry? 20 years?
At least.
Known Chris about that long too, so kind of feel like getting the band back together a little bit. But and Clint comes from Illinois. So we're going to kind of go through what Larry asked us to go through, figure out how to run this clicker. So who is Calibrated Agronomy? So it's a mix of integrated ag solutions, which is I'm part owner in at Denison, Iowa. I own it with Mike Wingrove and Kelly Garrett. Clint owns BioAg Management with his business partners, his brother, and Luke Holtz. And that guy on the end there holding that sugar beet, that's a sugar beet, is our friend from Idaho, Jared Cook. And we all kind of, I met Jared 4 or 5 years ago, got to know him. He introduced me to Clint, and we all became friends. We have very similar mindsets, a hunger to learn, hunger to figure stuff out, and we're very focused as a group. So why did we form Calibrated Agronomy? Most of the reason is we felt we were stronger together than apart. You know, running different businesses, different avenues. We want to kind of consolidate under one hood and try to provide some knowledge to the marketplace. So that's why we formed Calibrated Agronomy, and then also to help educate dealers basically is our motto going forward. So working with Larry's of the world and everything. But we kind of what we see in the marketplace is this kind of, call it you will, traditional agronomic model, you know, your chemistry, your fertilizer, guys you deal with every day, see kind of. And on the left is kind of what's the emerging markets, so like biologies, plant physiology, a lot of other stuff's coming. We're just trying to be in the middle, trying to bridge that gap for the growers and people that work in this environment. But really, our main motto is this at the bottom is what we've hung our hat on. The industry does a great job of treating symptoms. You know, the fungicide thing's a big thing, right? I mean, it's all saw southern rust, but so me coming from where I came from, you know, we go out and scout and see disease and you go spray fungicide. That was a reactive process, but why was that disease showing up is always the thing in the back of our heads. So that's what, there's always a root dysfunction when you see a symptom, right? You know, I got a headache, so I'm gonna give myself aspirin. Why do I have a headache? Well, there's a lot of reasons I can have a headache. Maybe I have too many drinks the night before or whatever, that's the key, or I hit my head against the wall or something like that. So what's the root cause? And that's what we try to, in our system, try to figure out. So we've also come up with this kind of this is something new to us. We've kind of been working on building this out this year through this last season of running through. And it's just what we're calling foundational agronomy pyramid. So it's kind of talking about what base things are at the bottom and we work towards the top, right? So preparation. You know, we'll talk about it today. Soil, or the soil, getting the soil right? Carbon, hydrogen, oxygen, structure, things like that. What we've learned is like, we still need to go back to the basics at times and fix things because all these fancy little products that show up on the market still ain't gonna change Mother Nature unless we work on it, right? Drainage, you got drainage issues, still gotta fix those. Things like that. So we'll work through this process today kind of as a format. And I'll let Clint talk about first thing.
Yeah. So Larry, Dee Dee, thanks for having us, and glad to be here today. Clint Frese from around Quincy, Illinois. Grew up on a farm, construction business, so put in a lot of field tile, ran a dirt scraper most summers as a kid, so very much into agriculture. Didn't go to college. I'm self-taught. My grandpa wanted to retire as I was getting out of high school, and he said, I'm either going to rent it out or you can follow me around for a year and take over. And so he's kind of the basis of how I got my passion for agriculture. And taking over a farm at 19, I made a ton of mistakes, was humbled pretty quickly. And so I've done a lot of things in ag on my own farm, and that's where it started. It really was out of curiosity. I was very research focused. and really big on data. And that's how I met Luke Holtz, and that's really what brought all of us together. But as growers, as farmers, we get this soil health talk all the time about how the plant is 96% carbon, hydrogen, and oxygen. And so as a farmer, what the heck does that mean? How do we manage carbon, hydrogen, and oxygen? And so what I really think it is, and it's something that we miss as we work with a lot of farms, and I think it's a very complex topic as we are transitioning from no-till cover crop or a more conventional system of tillage to a no-till system. We sometimes forget basic principles of soil. If you can't get a spade in the ground, you got a problem. And so for really what that means, how do we manage carbon, hydrogen, oxygen? It is, to me, management of oxygen and water in our soil. And if we get that foundational principle right and we manage our residue properly, and so to me, that's the sizing of residue, making sure our equipment can get through it the next spring. Those foundational principles are, to me, you get that right, and you've done a lot of things correctly. The fertility rate can vary greatly. A lot of things can change on the backside of that, but if this is right, I think a lot of things are good. So we're very much, as we grow, keep a spade with you. And if you can't get your shovel in the ground and then do some digging, how do you fix that? And sometimes it's not always what we've found is with a living root. So keeping that oxygen in the profile is big. So it's an example, a farmer we work with, he's got 2 farms. One he's managed for a long time. And so the one on your right is a field he's managed for a long time. The other one he's probably had 8 years, but very similar soil types. They're both pattern tiled, but one has great soil structure and one does not. And so as he went down this road of cover crops, min-till, he kind of did this whole operation. We're not going to do some way and some another. So the big takeaway was he had over a 30 bushel yield difference on these 2 fields with a cereal rye application. And so planted in the same type of time frame, similar management, fertility, everything. But the cover crop on the field with the very poor structure, this was a very wet spring, he turned that field into a swamp. And so the other field the field that kept oxygen in the profile, water was able to infiltrate. And so I show this because it's a great example of how when we get the carbon management wrong, we get the residue management wrong, it can not allow light to get down to let the soil dry out. It cannot allow wind to get down to help dry the soil out. So what we've done on his farm, we've reduced cover crop rates. We came in and fixed this with heavy tillage multiple times in dry falls. And we started over. We added some calcium, got this calcium-based saturation right. So there's things we can do, but jumping all in sometimes can be definitely left beating your head against the wall. So it's another example that I run into all the time, especially in flat geographies. And this field is also pattern tiled, so the farmer kind of has walked away from it. But what we have found is it's a super flat geography, and from walking it, he's got areas from probably back when they did deep tillage with a plow that are not allowing big pockets of this field to drain when we get heavy rains. And so this field needs surface drained in the worst way, and a lot of times that can be done with as simple as a tractor and blade. It don't have to be fancy or a dirt pan scraper. But as he's coming and fixed this was worth about 20 bushel on his overall field average. When we look at his historic tenure to what it is now. So I think it's just a good example of sometimes we can miss very basic things. But one thing we do and offer as a service, so all the NDVI maps now are free, Hyrdogen data, and so we'll do a lot of averaging of NDVI maps to look at historical trends and find these problem areas, go out and ground truth them and figure out what's wrong with them. But also we do this as we help farmers with on-farm research. We really like this system because it's not always, go to the next slide there, Mike. As we lay out farm trials, how many just a lot of times lay out farm trials on the fly? You turn the starter off, you switch hybrids here, or you put the additive in randomly halfway through the field. I'm guilty of that. I mean, that's kind of how I've laid out a lot of my trials in the past. But there's areas where maybe sometimes we create false data for ourselves by not knowing the historic yield, rhythm and flow and yield to our fields. So I really like this exercise as we think about in the off season, where should I lay out my farm trials? I'm going to do a hybrid trial, or I'm going to do a side dress trial or a fungicide trial, however you want to look at it. But depending on where we lay it could make that trial look really good or really poor. So one thing we offer is a cheap service is to come in and do this across all your acres. And then we can create management zones for VRT recs. But it also gives, I think, the next level approach to on-farm data management. So
Clint, let me barge in a minute here. So the last few weeks I've been doing some data analysis in our office, and we run across this all the time. And it's pretty frustrating, really, because, well, I know Ryan set up some planter trials, so looking at population, and we set blocks up like the Premier program does. And just so happened, where the blocks were, we did a high population and low in 2 different blocks and a check beside it. You know what the yield maps look like this fall? Well, it could have some of the best part of your field right next to some of the worst in a pothole. And that's kind of what happened. So it's a difficult thing to set these up. I like actually to do it more like you've got here and do a pass across the entire field. And for some of you guys that I've been doing that for, that's what I've tried to do. And even doing that, going one pass to the next pass on the combine, you see a lot of variability. So this is a tough thing to do.
it is.
And then if you have a in-furrow trial set up in the field, and well, then now it's time to put a foliar application on, make sure you don't get them on top of each other. Because pretty soon you got too many variables in that trial, and we really can't make heads or tails of it.
I'm guilty of all the above.
Yeah, I think everyone is, and it's tough to do, but it's something to definitely think about.
Yep.
So we'll go on to the next part of the foundational stuff when we talk about residue management. Residue management to us, so I'll admit I'm in a foreign territory, and you guys up here are flat. You got actually things called tillage tools. Where we're at Kelly's, we got hills, and you don't own a disc. So we're 100% no-till there, so residue to us is a little bit different than you guys. You guys obviously can turn it over. We have to manage what's on the surface. So residue has become a big deal to us up down there, but it's opened some paths. Talking with Clint in our group, we look at residue. We used to do residue as just getting ready for the planter and just recycling nutrients, kind of things you talk about. But this one avenue we've gone down is this disease management practice of the residue management piece. And we use this principle called competitive exclusion. So it's not a new term. It was actually coined in 1934 by a Russian biophysicist, I think biologist. But basically, what he found out is 2 species fighting for the same food source. He who has the most numbers wins. So it's a numbers game. So if I got 2 species in a petri dish, and I got a food source in there, whoever outpopulates the other one is going to win. So if I got a pathogen and a beneficial in that petri dish, I got to have more beneficials in that pathogen to outcompete it. So it's really the principle that talks about is the competitive exclusion.
I think the other thing within this competitive exclusion, it's one thing, right? Biology has a bad name. Biological products, bugs in the jug. If we don't know what our pathogen load is, and we're going to apply a species that's designed to say take out Pythium or Fusarium, and we don't apply the right rate, and the Pythium number is still above whatever beneficial species we're applying, we're never going to do anything with that product. Might as well not have applied it. So it's why we've resorted, and we'll go into it later, to more testing to understand. But when you look at this disease triangle, and you think about fields you struggle with disease, think back to this triangle. The environment. If you keep water and oxygen in your soil, you naturally stay in an aerobic environment. beneficial species want to thrive there. Our soils are not void of beneficial species. We can do more with changing the environment than we can with applications of crops. And so I'm very big on, you have to get the environment correct first, and your pathogen numbers automatically fall. So we spend a lot of time, we bury a lot of sensors. Soil Tech Wireless offers sensors we can bury to track ambient GDUs, soil GDUs, and moisture levels within the soil profile. And the interesting thing is, the wetter the soil zone is, the more pathogens there is. And so it's goes back to, I think, very much common sense, but the disease triangle, when you're working through problems on your own farm, I think there's a lot that can be taken away from just going back to the principle. And then, making your plants a non-host, you got to get better at mineral nutrition. The better job we do at balancing our plant nutritionally, the better job it does at fighting pathogens. So it's, I think, a good system. If you're struggling with disease, you've probably got an environment problem, or you're making your plant a host. And so if you over apply nitrogen by 100 pounds too much, probably making your plant a host. If you got drainage issues, compaction issues, you probably have your environment is switching to an anaerobic environment that then breeds pathogens. So some basic things I think that's important to principles to hit on. So
You got something there?
Well, I just, one of the things I did more this spring than I ever have in the past is, this is a soil penetrometer, and we've had questions, different tillage systems and things. And with all you see all the pattern tile drainage that's been put in the last 10 years in our area here. We used to, and we still do a lot of tillage. We turn a lot of soil over and turn a lot of ground black. I think actually, I hate to say it, but I think we're overdoing tillage, and we could do with a lot less. And there's a lot of money spent on tillage. Those rippers and things are not cheap implements to keep maintained and keep running, and also the big horsepower to pull it. But so this spring, I did a lot of testing with this. The ideal range to be at is somewhere between 100 and 150 pounds to push this down through the soil, up to as far as this goes. And we were nervous on some, I had a farmer that's in his 5th year of this season of no-till, and was just nervous that we had compaction issues, and especially in the tracks where the planter goes and where the sprayer goes, he tries to control traffic. And this spring he had the field cultivator out, and we went out to those fields to look both in bean stubble and corn stalks to see what the compaction if there was compaction out there and where it was. And I think both of our surprises somewhat, this thing you'd push it down through the ground and it'd be between 100 and 150 every time we pushed it in, even in the tracks. So we were in good shape. We thought we'd have issues, but we really didn't. So I guess in all and again, we've needed tillage in the past, historically, because we needed to dry things out for spring. And it definitely does do that, but I question whether we do that more than we need to. So you can find fields in conventional tillage where you definitely find, and I went right across the road to neighbors that did conventional tillage, and sure enough, about, 8 to 10 inches, you're going to find a layer, and there's definitely layers there that you're dealing with. But if we stand long enough and do the no-till, I think it's a valid thing we could do. As far as residue management, you maybe get into this a little more. If we do less tillage, I think it's more important to use a biological or something to break down that biomass, because again, I think you'll show later on, you'll see what diseases can be carried over on that residue. So that does become an issue. But even if you're tilling that stuff in. I just wanted to add that. And a lot of guys are doing this vertical tillage now as well. I'm not against the vertical tillage so much, but yet in doing so, you're mixing in that residue. If you have a residue manager on and you do vertical tillage, you're probably okay. But if you don't get that residue manager on, now you've got all your 200 plus bushel corn residue sitting in that top few inches, and you're planting into that. So a clean furrow is an important thing to have as well. So I think you'll actually have a better furrow in a no-till situation than you would in a vertical till situation.
Well, yeah, it's like our no-till, like residue management behind the combines, very important.
Yeah,
I have a bunch of guys who have 40-foot heads but can't throw 40-foot combine.
that's a big issue
Or don't
too.
change it. And then you got this wad right behind the combine and you're trying to plant through it and you just, we had one year where the combine messed up on one field of Kelly, so we had a 15-inch planter with row cleaners on it. You want to talk about an upset planter operator? We couldn't get through it. It just wound up. So, I mean, running the combine is, all the other things you got to do, residue management on the combine is critically important too.
Yeah. Very much so, especially with bigger machines and the wider heads. I remember back in the 80s, when we had the first 9600s and we had a 30-foot platform on, we thought, you know, but I can remember time and time again, you couldn't get that residue spread out that 30 feet, and we had issues next spring. You could see that in the cornfield, it's just a wave cornfield the next spring.
So this is some data from, we partnered with Earth Optics. They're formerly, they're a merged company from Pattern Ag and Earth Optics. So they took on the Earth Optics name. So we've been working with them, what, 4 years probably? Yeah. So this is from their this year's report from last season. So basically you're looking, your left is corn, right is soybeans. And what they did on like 30,000 acres, they took the soil data versus yield and they found the highest risk what was affecting. So they'll measure what they call crop protection and other things, but basically pest and disease is the orange circles, your biofertility, so your biofertility colonies, how well your biofertility is.
Mike, explain a little better what they're doing. They're doing like a DNA test.
Yeah, so they basically take a soil sample like you would your soil fertility, send it to a lab and they DNA sequence it. So they're basically scanning that soil profile against the metagenomics database. So there's, I don't know how much is in there, but it's a well-known database all the companies use. They just reference that. And it could be living, right? It's just scanning the DNA. So it's living, dead, dormant, whatever is in your soil. And basically what you get back is a report, and it's just a risk factor, right? Look at it as risk factors. Just because that number's high doesn't mean it's going to show up 100%. It just means that you have a higher probability if you're going to have fusarium, per se. If you have high fusarium, you have a higher risk for that to show up in your crops. Now there's ways to mitigate that, but that's how you got to look at it. It measures, and they keep adding stuff. Nematodes are starting to show up in there now. Pythiums, rhizoctoniums, and foliars, tar spot, and corn. I mean, they've got a wide database they're starting to keep continuing on add to. So that's what they do, and that's what we like. We're doing more of that with guys as we roll forward. And we use it as a testing platform for products too. We'll show that here in a minute. But this chart basically showing your highest risk factors of yield loss based on their data set. And if you want to look at it later, I can show you, but basically, at the end of the day, what I wanted to point to is over 50% of what's up here on your highest risk involves or diseases or lack of biofertility pathways that are involved in seedling growth and just seed germination. So it's pathogenic loads in your soil that are going to affect your seed growing out of the ground. So when we're talking about residue management and everything else, that's, it's important to know what you're dealing with. I mean, we've got a lot of guys here lately that have good soil fertility levels, done a good job, but their yields are just stagnant. And we're starting to look at this as another option, as another layer of data in their fields to look at. Where's your soil biology at as far as pathogenic load or biofertility pathway? This is about an $8 an acre spend to get these per field. So
I think for farmers on more limited budgets, you can pull this in composites. Pull your wet area, pull your hilltop, pull your different soils, your demographics, and it'll give you a baseline look to start. So,
Yeah, that's $120 a sample, so, to do that. So then, going with this competitive exclusion on your left is another study they showed us just on Fusarium. So the green side of that is low pressure fields, and you'll see the yield go up. So the X and Y axis, I'm very good at graphs, but the top and bottom is your yield, and all those data points are field locations there. On the right is your high pressure fields and all the data points. It ends up being about 25, 30 bushel in their data set for high risk Fusarium field versus low risk. So how do we change that out? Well, with Clint, I'm doing the testing with BioID, and you can see on the right side of this chart is those are long-term fields. On the left, your right, excuse me, be the untreated, and where we're treated with our biology, we've lowered that risk on those fields. So just looking at how we can mitigate the competitive, or use the competitive exclusion to mitigate that disease risk.
in a soil ecosystem, you can never drive out the pathogen 100%, but you can outcompete it and lower it to where it becomes unplanned and infectious. And so that is our goal as a company, really, is to try to how can we work with nature and actually measure what we do. And you know, our product BioAggie, we're constantly trying to shove more in it. And so BioAggie is really, it's a diverse biological. It is not a rifled approach. And so when we first started, I really was focused on what biology did to nutrition. But I think as a farmer, to keep things as simple as possible, in our soil, biology is responsible for delivering nutrients. It's very responsible in carbon fixation and how the plant uses carbon. It's very responsible in what they call biocontrol, beneficials that compete with pathogens. The other two big ones is biology produces all of our growth hormones that our plants need to grow. The last functionality group within the soil is your stress adaptation. So that goes biology that helps create aggregate structure, exopolysaccharide production that creates biotic films to section the root system from heavy metals, salts, and it's a protection mechanism for the root. But they also, in the stress adaptation pathways, produce all of the defensive hormones that a plant uses to protect itself naturally against pathogens, as well as senesce the plant as it's finished in its life cycle. So biology really is controlling so much, and what we tried to do with BioAggie is put everything in there to get all functional groups. What we believe is there's 1 billion species in the soil, and to go out with 1 or 2 species is a really tough ask, especially if you don't really know what's your limiting factor. And so what we've seen a lot is, you got a pea solubilizing bacteria that is marketed as, reduce your phosphorus by 50%. Well, if your soil already has really good pea solubilizing pathways, you probably can reduce phosphorus without that product benefit. So I think with the one thing the Earth Optics testing does, it brings everybody to a playing field to where we can look at, hey, where am I at currently without any products or stimulation, and how can I use products and actually measure what I do. And it don't have to be products, it can be management. So maybe I change rotation, maybe I change to a rotation grazing or a diverse cover crop blend. You can measure change over time as you do this. And the other big one we've used it with farmers is eliminating where we come in fixed compaction, we get drainage correct. Pathogen load naturally lowers on its own. No products applied. So it's been a great learning tool really for us.
So let's compare the biological, BioAggie, let's say,
Yep.
or other ones that we've used in the past, to the synthetic products, that the Olivos, the Saltros, the things like that. Let's compare that. What do you see with that plant?
Okay, so when we use fungicides or synthetics, there's almost always For every benefit, there's a negative that coexists. So what I have found with biology, so I had a farmer we worked with for quite a while. He's been on our BioAggie and furrow program, likes it, really is eliminating rooting diseases from his farm. Picked up some new ground. He used the BioAggie. We had a crazy wet spring, some 4 and 5 inch rains. The new farm he picked up, even though he used the BioAggie, it got hit with Fusarium and Pythium bad. Had to replant his new farms. But he applied the beneficial biology that should counteract Pythium and Fusarium. Biology takes time, and the reason I tell that story is you go out and apply a product once, and you say, well, that didn't work. What we are more after is how do we change biology in our soils over time. So what we found is biology isn't sexy. It isn't quick like a synthetic. If you got a disease and you want to kill it now, use a fungicide. If you want to work for a long-term goal of driving out Pythium and Fusarium, Rhizot, use biology, and I guarantee you over a 2 or 3 year time frame, focus on water and oxygen in your soil, get those species built up, you won't have that problem on your farm. But it's a case in point of, one's very quick, Larry, like for the synthetic, or the biology, we find by year 2 it should be ready for you. So, but this is when you get back and you log into the Earth Optics portal, I think they do a nice job of just color coding stuff. So how we have validated products or different management practices, when you see a red graph, so Fusarium is in the red in the top there, that means there's a high likelihood that it's going to be plant infectious, and especially if there is a hybrid or variety that is extra sensitive to Fusarium. If it's in that orange area, it can go either way. Depending on the environment, we're not as wet. We naturally have a little more oxygen in our soil. Where it's orange probably could go either way, where maybe isn't a problem, or if we go crazy wet, our environment shifts, probably going to be a problem. When we get to the green levels, that's what we're really looking for with our pathogen loads. We want to keep those where they have a very low likelihood of blowing up in the soil and becoming plant infectious. So this has been an eye-opener for us, and really why we've built back in the foundational pieces of agronomy of water and oxygen management in the soil as such a crucial piece to our pathogen.
So how often, how frequently are you testing for the Earth Optics? How do you do that every couple of years?
On composites or research farms, we're doing it quite often. Like on our research sites, we're doing this about every couple of weeks to get to understand the rhythm and flow. And that's where we've learned that as Mother Nature dumps 5 inch rain on us or rains are more timely, it has the greatest impact on this. But as a whole, most of our farmers are every 3 to 4 years. Really trying to track change over time and keep it cost effective. So,
yeah, I'd say probably every 2 years. Be my minimum on time, guys. The Earth Optics, obviously, they're a lab. They like to get samples, so they like to do it every year, just to, especially if you're rotating. If you're monocropping, like corn on corn, you can probably get away with it every few years. It does like log root worms and soybean cysts and stuff like that. They give you that, so those things will come through.
Are you taking and using a residue digester to help offset pathogens?
yes. So probably our biggest, Jared Cook works in Idaho, more specialty crops, but we've been doing some university work and working on really PhD research on trying to get rid of fumigation in vegetables. So onions, sugar beets, potatoes, fumigation's used in a lot. And so what we found is year 1 as we was trying to replace fumigation, where we didn't come in with a fall program and start to outcompete pathogens where they are harbored in our residue. We only had a success rate of about 80% driving pathogens out of, say, onions year 1, where we come back year 2 with a little different program and started on the residue in the fall and then went on the seed, went in furrow. That's where we're getting 99, 100% control pathogens. And so it's a season-long approach, but we're probably, like in a lot of our vegetables we're working with, we're spending $60 an acre on biology from fall to when they harvest, but they're spending $400 on fumigation. So, I mean, it's a pretty big offset, but there's a lot of the processors in our food system right now. They're really searching for alternative ways to get around some of these intensive, fumigants and high applications of fungicides. So the consumer's driving that, and so they're starting to do a lot more research with universities and with biology and nutrition to try to offset some of the tools. It's pretty standard in vegetable and fruit and application. So, good question. So this is like a list that we have from BioAgE, and this was a 2,000 acre, 1,600 acre study, I believe, where we applied BioAgE for in furrow for 2, in some fields 3 growing season in the pathogen reduction that we encountered. So quite a few diseases and nematodes that we affect and drive out, and so that has been what we have based a lot of our data on, is looking at larger data sets, working with different agronomists to kind of vet this out over and over again. So it's very real, and you don't have to have a product be claimed as a biofungicide. We're not a biofungicide, not labeled as one, but you can still do these exercises on your own farm and really understand how you can affect pathogens over time. So
when we look at, to Jake's question about using residues as a disease suppressor, so to speak, up on the left of the screen is the cycle of tar spot in corn. So what we've come to find, these diseases harbor in the residue in the soil, and some of them can sit in the soil. White mold's a good example if you ever dealt with white mold. There's a bunch of research, even from like Fair and other places, talking about white mold living in the soil for up to 5 years before it becomes prevalent. It can re-spore out. It can live in dormancy for 5 years in your soil and then show up again. So they harbor in the soil, harbor in the residue, and you get this cycle of growing the plant, harvest it, you got disease, put it back on the ground, start the next year, you already got the disease laying on the ground, and it just cycles. So we use like a version of BioAgE we call breakdown to implement into that cycle to break that, to eliminate the host. So you go back to the disease triangle, the residue is a host for the next host, right? So we can break that down, get that accelerated, we can lower our chances of having it next year. So here's one, this is last winter, we had a meeting in Laurel, Nebraska, kind of an interesting area, I think 90% of that is irrigated country, a lot of corn on corn, mostly min-till, strip-till, no-till. But we went out to one of these customers, and Jared found this actually just loaded with tar spot residue still in February. I mean, that field looked like it just got combined. The grower himself is concerned to even plant next year. So, these visuals, you see it in the season, you're going to have it in the fall. And using residue digesters, like with Extreme Ag and everything we do with Kelly, we were trying to cycle nutrients. Now it's become like, let's manage the disease for us as another tool to get this on. This is your study of it.
yeah, this is we worked with an organization called Novus. Got a lot of different sites, but this was a replicated study where the interns went out and just did a hula hoop test. And this is no-till, slight min-till ground. And so I think from November application to April when they did hula hoop ring throws, we reduced they came up with like 1.37 ton less, and really cycled more nutrients there. But back to one of the fields they pulled it from, you can see the hog buildings in that picture. It's kind of hard to see. But where we seem to have yield benefits from fall applications or in that manure environment, think about plant biomass. The bigger the plant biomass, usually the less air movement we have in a crop, usually the more disease we end up with at I guess senescence or harvest. And so more disease spores on the residue. But this is an example. I think we had about 5 bushel in this field. But we don't always see this. So if you're in heavy tillage, you know, I don't think that maybe this is as important. When you oxidize your environment, your residue, you create crazy biological activity that digests your residue. So if you are in a no-till, that's where we see this shine. The other spot we really see it shine is really the high disease load fields.
So we probably CFS North Line.
The long-term study we've done with several farmers, and it's an organic matter study. So think about biology in our soil and what it takes to create stable humus. And when we've applied bioag into a lot of different soil environments, we do see an OM jump. And so this was back, I believe, 2019. When we sampled this in 23, no change from where we gained. So probably going to take more carbon input from here to continue to increase the organic matter. But I think the reason why we show this is there is species in there that will complete that carbon cycle all the way to stable organic matter. This is from some work we did in the river bottoms, and so all sorts of species that increase aggregate structure, aggregate stability. This was a long-term check. We had a farmer that we was working with, and he left basically that's 4 years untreated versus 4 years treated. And he made changes to his operation at the same time. I mean, he was going more min-till, strip-till at the same time. So, but impressive differences. Just a water check we did in that same picture, just holding our soil together better. Pictures from after we apply in furrow, a lot of times we do have a very high fungal count, and so we'll start to see bigger aggregates being built on those fungal type clusters, and bacterial then will feed and work in those aggregate areas. So one thing that we spend a lot of time doing is looking at fertility curves. So I bought in, when I first got into biology, started playing with it in 13, I bought into the whole, you don't need fertilizer anymore. And so I went almost 10 years on my own farm and some of the research farm that we was renting without fertility. And so I was basically managing nitrogen and using biology in furrow and walking away. And so we're blessed where I'm from in West Central Illinois with some pretty good ground, but then we go to really timber soil, high clay content, rolling hill type ground. And so we spent a lot of time, I always blew strips of fertilizer in all my fields every year over this 10-year period of when I reduced my fertility. And we had a lot of customers that we reduced fertility by 50%, and we would come in and soil test every year. But what we found a sweet spot was, as we reduced off of U of I rate Rex by almost 50%, we never really ever seen a yield decline or a fertility change as we brought the biology in. And so what we did see though is on our heavy dirt, no really PK added. I didn't see major yield ramifications. We still continued to crank out 240 corn, 80 bushel beans. But my light dirt by year 3 and 4 was trending below my 10-year average right where I pulled out PK. So no fertility is not the answer, so the answer is usually somewhere in the middle. But I do think we do have resilience year to year with fertility applications depending on price. So
that's something we've done over the past few years when fertility prices or fertilizer prices have gone quite high. In fact, the last 10 years probably, I've actually started decreasing fertilizer rates in the fall. And I don't think we can say that we've actually lost yield to that. Can anybody disagree with me? I mean, how much of what we put on is actually available that next year anyway, first of all, and we're still maintaining our fertility levels when we do our soil tests. So this past season, I don't know why, but we see that in soil testing. This past season, I've seen more lower levels than we have in a long time. We've seen potassium levels drop significantly on some fields. I can't explain it. We haven't really changed anything. We've been putting on potash. In most cases, we're putting on potash at least removal rate, and you know, some cases where we're really high levels, we've gone less than we're putting some on almost everywhere every year. And why those levels came down this season, I can't quite explain. That
Was it fall cold?
no, we're doing most of our sampling in the spring after planting. But the trends you kind of see going up and down. It's not a perfect science, the soil testing, but so yeah, I just wanted to add that. I mean, we've done less than removal in many cases over the past 10 years now, and not really seen reductions.
But it is something to monitor, and I
Yeah.
think the end goal, though, is to, and what we're gonna go into, is why we went to letting the plant tell the story. Because fertility, as we are learning more about biology and how the plant is delivered nutrition through the rhizophagy cycle, something we didn't know 6-7 years ago. I think it's getting more complex about how nutrition is actually delivered. So I can go to the next one. So this, I always find this one fun. I always tell everybody, give me all year to pull your soil test and I'll make this test look like however you want. And so in season, a lot of times we do have a much higher test, and it's good that, Larry, you are testing on the planter. It's probably the most accurate representation you can have. So if I see what a lot of things that I see trends on, I get a lot of phone calls from guys that are on a fall testing program, and samplers will go ahead and pull on a dry fall, and they look really, really poor when they've been on a very strong fertility program, and it's very much a false test. And so when you pull matters, I guess. So
Another thing that just came to mind, we were pretty dry last fall.
yep.
So when we're pulling samples in May, maybe we weren't totally cycled yet. I don't know.
It could be. I mean, if the subsoil is drier, it will have a false read, is from what I've found.
That could be part of it.
So, a lot of claims with biology and how much you can reduce your fertility. It is a year-to-year variance, and it goes back to environment. If the environment is allowing biology to thrive, the reduction in fertility can be great. If the environment is not allowing the biology to thrive, the reduction is little. And so we spent a lot of time in replicated plots measuring before we started this app. We looked at tissue here in the green and just the increase in nutrient flow from B4 to tassel. Bioag applied, a biological applied, and what was the increase in fertility to the plant. And so this is something we learned from Next Level Ag Lab. And anytime you want to evaluate a program, whether it's a nutritional program, a biological program, you basically, what we do is we come and we look at a control plant, nothing's been applied to it, and a treated plant. And we pull the same number of leaves in both, and you weigh those. So you'll weigh them in your tissue bags, and when you get your tissue reports back, you either take one number up by the percentage that it weighs more or one number down. But it takes plant biomass into effect, and it will allow you to evaluate how much more flow or how much more nutrient is stored in that plant due to the increase in biomass. Tissue test with the dry combustion test, it does not take plant biomass into effect. So whenever you want to evaluate how much more FOS is this product giving me, or how much more is my starter sped up nutrition to the plant, you can do some of these exercises. And probably the most eye-opening thing, we was working with Next Level, and we looked at a biological plot at B6, and he ranked my plot from 1 to 10 at B6 on yield. But we had the biomass and we had the tissue results. So there's some things we can do with sap to correlate to yield now, and as well as tissue. So
Manure.
manure. So yeah, so one of the areas that we are growing fast in is we put a lot of our biology in pits. Basically turns, we look at it as a free application zone, and we spend a lot of time starting to look at the pathogen load reduction in the manure, but what we've really been focused on is first year availability of nutrients. And so as a whole, we really speed up first year availability in the nutrition of the manure, and usually see a nice yield benefit from that. Also, we get a lot of comments about less solids floating, and we seem to get our pits more empty. So I think we're doing a better job with the soluble control of solids as we introduce kind of a different spectrum of biology into our manure pits, because manure pits are loaded with beneficials and pathogenic biology all at the same time. So